For our research proposal, we're looking at the presence of Escherichia coli, or E. coli, in stagnant versus running water sources. We noticed that in many cases around the country and world, there are substantial issues with obtaining safe, drinkable water. For instance, the Flint water crisis is a prime example in which the citizens' main source of water was contaminated with lead and deemed unsafe. The U.S. Environmental Protection Agency found that nearly half of our rivers and streams are polluted, forcing us to turn to other sources. To locate these alternative sources, we decided to test different water sources for microbial growth, specifically E. coli. Through our previous research, we learned that E. coli is a fecal indicator bacteria that is a major cause of water contamination in the U.S. This harmful fecal coliform can lead to diseases such as diarrhea, gastroenteritis, typhoid fever, and salmonella poisoning. For this reason, it serves as a recognized indicator bacteria for contamination.
In our research, we will be testing the question, how does the presence of E. coli differ in stagnant water versus running water? For our stagnant water sources, we will be using 2 ponds and 2 lakes, and for our running water sources, we will be using 2 streams and 2 rivers. Our null hypothesis is that the presence of E. coli is not affected by the water source, and our alternative hypothesis is that the presence of E. coli is affected by the water source. We would like to use the findings obtained from this experiment to determine which water sources are the safest for consumption and could serve as low-cost substitute sources. Testing serves as the first step for adopting efficient water treatment methods in order to reduce the transmission of pathogens. As we discovered in our previous research, E. coli is 26% higher in contaminated water than inadequately treated water. Overall, our data could be vital in finding viable sources of safe, clean drinking water in unsafe water areas such as Flint.
All right, for our experimental design, we have 2 different types of water sources, stagnant and flowing. These different collection sites were chosen because they are relatively accessible sources that people can reach if they have contaminated or unsafe drinking water. E. coli was chosen since it's a very common contaminant in drinking water according to the guidelines for drinking water quality. When we decided to test velocity, since we wanted to see if there's a visible difference with stagnant versus flowing water sources and if that was measurable with velocity. Finally, after reading from Jagupilla et al., it should be a point to try and get all tests after a dry day or a wet day, just to ensure a controlled experiment, which is why we also want to try and get our tests reasonably around the same time. For our variables, our independent variables are going to be our water source and water flow. And for our dependent variables, it will be the velocity of the water as well as the E. coli presence. And if you look at the chart on the right, you can see our different water sources flowing and stagnant and our different selected rivers, streams, lakes, and ponds. For our methods diagram, we will start by showing up to our water source and figuring out the velocity. We will do that by dropping a leaf at the 0 meter mark and finish timing it at the 5 meter mark to get our velocity. And we will also be getting a water sample at the 5 meter mark. Once we get our water sample, we will put 10 milliliters into one of our testing tubes, which has a testing tablet with the chemicals in it. We will store the samples in a room temperature area, dark area, for 24 to 48 hours. And once that testing time is up, we will shine a UV light to see if it is positive or negative. If it is a positive E. coli presence, there will be a yellowish glow at the top of the testing materials, and then if it is negative, it will be a reddish glow at the bottom of the testing materials.
To analyze the data that we are going to collect from the 4 different water sources, as well as the velocity from the 4 water sources, we're going to use 2 different statistical tests. The first test that we're going to use is the chi-squared goodness of fit test with variables of the presence of E. coli and the water type. We chose this test so that we can measure or look at the correlation between the presence of E. coli as well as the body of water type to see if the 2 are dependent on one another. The other test that we're using to analyze our data that we collect is a t-test that will examine both the velocity of the water as well as the presence of E. coli. We chose this statistical test to determine whether or not there is a significant relationship between the velocity that the water is traveling and the presence of E. coli in the 4 different bodies of water. All of the data that we're going to collect will be analyzed using Vasterstats to compute both the chi-square goodness of fit test as well as the t-test results. For our resulting data analysis, we determined that the acceptable p-value to reject our null hypothesis will be any p-value that is less than 0.05. If we do have a value that's less than 0.05, we can then reject our null hypothesis and accept our alternative. For our two-tailed t-test, we said that any value of t that is greater than 1 will be able to reject our null hypothesis and accept our alternative hypothesis. For our timeline, we're planning on having one data collection date where we can collect our water samples as well as measure the velocity of the water. After we've done that, we can record our velocity data. We can then prepare our E. coli tests, and after 24 to 48 hours, we'll be able to record our results for the presence of E. coli. To get the results of our presence of E. coli test, we can then begin the statistical testing so that we can work on our final presentation. For the project, we used the Lyman total coliform and E. coli bacterial test. We used 2 of these. However, if we were able to have a larger budget, we would collect more samples to get more accurate results in the end. Here are references just in case anyone would like to look into our project or get any more information about what we're looking into. Thank you for listening.